So in today's lecture, we're going to be looking at how DNA is replicated, as well as how bacteria are involved in gene regulation, especially transcription. That is going to be the first thing that we're going to be looking at. So as you know, the cells that are dividing are going to replicate their DNA. And in order for that information to be accurately passed to their offspring, the chromosome needs to be duplicated in such a way as to maintain all that information intact. So what you have learned from before is that DNA replication happens in a semi-conservative fashion. What would that mean is that a mother part of DNA, this DNA molecule, which is going to be the one that is going to be duplicated, it's going to maintain one strand of that molecule in each of the daughter strands. There's another type of duplication in which now you have the mother and an identical replica of two daughters, but that's not the way in which DNA is replicated. So that's why we call it semi-conservative. So semi-conservative DNA, as you know, it has happened in all the organisms, and it involves copying both strands to generate an identical copy of them. Now, as you know, replication happens in the 5' to 3' direction. And that's going to be important because it's going to give us an indication of the mechanics of replicating a molecule that has two antisense strands. One replication, as you know, is going to happen sequentially. That's the leading strand. And the other one is going to be interrupted into small sequence pieces, and that's going to be the lagging strands. So, as shown over here, for adding a new nucleotide based on the sequence that the polymerase is reading, you require a hydroxyl group that is free at the 3' position. And what I did was that I circled in orange over here the fact that this is going to be a condensation reaction. You're going to generate a water molecule, and the hydrogen from the hydroxyl group of the sugar and the OH group from the phosphate, the third phosphate in the nucleotide, will be taken together to generate the phosphodiester bond that is going to add the new nucleotide in place. So you have, again, the 3' hydroxyl from one side, you have the 5' phosphate of the other molecule coming up, in this case from the new nucleotide that is going to be added. You're going to generate a pyrophosphate molecule. That's the principle about the sequencing that we've been discussing for the pyrosequencing, having that pyrophosphate be generated, and that is going to be released. The pyrophosphate molecule, however, needs to be hydrolyzed by the enzyme pyrophosphatase, and what that does is basically make the elongation reaction irreversible, because you do not have now another pyrophosphate to add back. So the cell is going to also be looking at the concentration of pyrophosphate. So degrading the pyrophosphate helps prevent the enzyme from now performing the reaction backwards and eliminating the nucleotides being added. That is actually one quality of the enzyme that we're going to be looking at, and that is the 3' to 5' exonuclease activity. So as you know, the deoxynucleotides are going to be the substrate added for the molecule, especially when you're talking about DNA. And you're taking a triphosphate molecule and adding a monophosphate molecule into the elongation reaction. So the question always comes, if you have a double-stranded DNA molecule, how can you start replication if you're lacking a 3' hydroxyl group? And that is the purpose of having the enzyme primase. So primase is a DNA-dependent RNA polymerase. It's going to add a random 11 base RNA primer into the sequence, and that primer now provides the 3' hydroxyl group needed to begin the elongation of the chain. And as it says over here, it's found in all the organisms. So here in this image, you have the single-stranded DNA in light green. And as you can see, you begin the enzyme primase to add an RNA primer from the 5' moving to the 3' direction, about 11 bases, and after that the DNA polymerase is able now to add nucleotides to it. So what we're going to learn is that in eukaryotes, DNA polymerase 3, it's the enzyme that is going to begin the elongation of the new strand. In a circular chromosome, you have a single origin of replication that is going to be recognized by the DNA replication machinery. And you're going to create a replication fork that, as you know, is going to go in two directions. One over there to the left and one over there to the right. Each replication fork is going to have two polymerases. So at the origin of replication, when that opens up, you're going to eventually have 4 polymerases into 2 independent complexes that are going to replicate DNA forward. And as you can appreciate over here, the origin of replication is shown in pink. You generate this traditionally looking theta structure that is known as the structure that it's traditionally seen as evidence of DNA replication in prokaryotes, because it's the elongation of another circular chromosome coming out. And you'll have, in a chromosome, as it's separating, one strand will have a leading strand and a lagging strand together. So you can appreciate here at the bottom that there is the replication fork here in the left has a leading strand here in the bottom and a lagging strand here on the top. The replication fork here on the right has a leading strand in the front and a leading strand at the bottom and a lagging strand on the front. So they are placed differently because one molecule is going 3' to 5' and the lower molecule is going 5' to 3'. So that switches which of the strand is the leading and the lagging. So you can appreciate that. So keep an eye on that. Now the other thing that I want to remind you is that the origin of replication is a DNA sequence. I'm going to take a moment to look at that sequence in more detail in a moment. But first, what I would like to do is to go over with you all the genes that are encoding proteins involved in replication. Now, you're going to have DNA gyrase. This is a list, so let's just go quickly through the list. DNA gyrase is encoded by the GAR-AB gene, and as you know, that is going to be the protein that is going to unwind the supercoils. So you're going to release them. You're also going to have a small protein called origin binding protein, and that is the product of the gene DNA-A. That protein is going to form a complex in which the origin replication is going to wrap itself around and melt the strands. I'm going to show you that in a moment. Helicase, it's an enzyme that is going to help separate the strands to allow the replication fork to move forward, but it needs to be loaded. And the helicase loader, the product of the DNA-C, it's going to be that enzyme. So the helicase-C is going to help load the helicase, and helicase is encoded by the helicase, the DNA-B gene. We're going to look at helicase in a moment too, because it's actually going to be in the lagging strand moving forward, and usually associates with the primase. So they're together. You also have the product of the SSB gene, which are the single-stranded binding proteins. As the origin of replication melts, you do not want it to reanneal back together. So those small DNA binding proteins bind and sequester the capacity of the DNA to reanneal back into a unit. DNA-G gives you the primase, so that's going to add the RNA primase that we talked about. The polymerase, DNA polymerase 3, it's a multi-unit polymerase, and it has a clamp that is going to be the sliding clamp, that's the DNA-A. The clamp needs to be loaded, so you need the whole A to E clamp. You have DNA-X, which is going to allow that unit to dimerize, so therefore you can have 2 polymerases placed in one area. Of course, the DNA polymerase unit per se, that is the DNA-E. And then you have the proofreading unit, that is the DNA-Q. The proofreading unit is the one that allows the 3' to 5' exonuclease activity to correct mistakes when the DNA has been replicated. A very different polymerase, it's Pol1. And Pol1 is encoded only by one gene, Pol-A. That is the polymerase that is going to remove one by one the bases from the primer and fill in now the oxynucleotides to replace that RNA with DNA. However, it cannot fix gaps. That is going to be ligase. And ligase is an enzyme with two proteins, Lig A and Lig B. Now, at the very bottom, remember from the structure of the chromosome, you have a termination sequence. The termination sequence is going to be bound by the TUS protein. And that is going to prevent the continuous replication of the chromosome around the circle. It just goes from the top to the bottom and stops. And last but not least, topoisomerase 4. Topoisomerase 4 is a type 2 topoisomerase. It does double-stranded DNA breaks. It's not the same thing as the topo 2 that we talked about yesterday. But it was a bit confusing when I gave the lecture that it sounded like it were the same molecule. Topo 4 is the one that separates the circles. Somebody's bringing cookies. Topo 2 gyrase, it's the one double-stranded DNA, inducing double-stranded DNA breaks. Topo 4 is a type 2 topoisomerase like gyrase, also induce double-stranded DNA breaks, but it's required to separate the circles. Right. So that's a clarification that I wanted to make for you. So let's take a look at how the DNA gets replicated. And let's talk about DNA A, which is going to be the origin binding protein. Now, that forms, when you look at the origin has 4 9-base repeats. I'm going to show you a picture of that in a moment. And the product of DNA A binds to that area. That is going to induce a conformational change in the DNA downstream in an AT-rich region. And that is what's going to melt the origin replication to allow replication to begin. Now, you have between 10 to 20 monomers of DNA A, and they also require ATP to do this. So you have that ATP-rich region with 13 bases. The AT-rich region has 13 bases in each of them, and they're intertwined down the origin replication. I'm going to show you a picture of that, so hold on. And when DNA A binds to the 9 base pair repeats, that is the part that melts away. Once it melts away, that is going to guide the helicase and the helicase loading complex into the replication fork to allow now helicase to start doing its job. So let me show you now this in a picture. So here it's an image depicting the origin of replication. You have these 4 9-base repeats. The arrow indicates the directionality of them, where the repeat is moving. And here they're shown in blue. And you have these 3 13 base pair repeats which are AT-rich. As you remember, A and T's have only 2 hydrogen bonds. Therefore, the strength of those AT-rich regions is a lot weaker. So here you have now DNA A as a complex, and as you can see, it's wrapping around the 9 base repeats which are in blue. When that wrapping around happens, that is going to now melt the strand and separate the AT-rich. So now 3 things are going to happen in here. You separate the AT-rich region. You also allow the single-stranded DNA binding proteins shown by these dots to bind. So now they attack, and they have now prevented that region from binding back together. Yes? That's what we always, when we use the term melting, is how we refer to separating the 2 strands. No. It's not an indication of depth on energy. Thank you. So as the AT-rich region melts and separates, the single-stranded DNA binding protein can come in and now bind to the single-stranded DNA. That prevents it from annealing back together. And now you have the helicase and helicase loader as a complex. So the DNA B and the DNA C, 2 of them, because you have 2 replication forks. So one of them is going to load in one strand, and the other one is going to load on the other strand. From this image, notice that the helicase is not binding double-stranded DNA. The helicase is binding to a single strand. We're going to look at that in a second. Okay? So that wrapping, and this is all that information there right now. So I said it all, I'm not going to repeat it then. So part of the thing is that the orientation, the spacing, and the sequences of those 9 base pair repeats are critical to the function of the origin of replication. The ATP richness of the 13 base pair repeats are also crucial to the function of them. So in total, this region is about 245 base pairs long. And it has those 4 9 base repeats, which are the blue, and the 3 13 base repeats, which are the red ones. So now you understand how the origin replication is required to initiate the replication of the DNA. So you have a physical separation of the strands by binding DNA A. Now, as I showed you earlier, helicase, it's binding to only one strand. It's binding from the strand that from the right to the left is going 3' to 5'. So that is the strand that is binding over there. So here you have an image of helicase. Helicase is an ATP binding protein and is going to use ATP to mediate the turning of it. It has a 5' to 3' helicase activity, so it's moving from the 5', moving to this side, to the 3'. So that's why it's always moving into the direction of the double helix. And that's why it can separate them. So you always know in which direction helicase is loaded because it's moving 5' to 3'. So think about which strand is going 5' to 3' in the direction of the fork, and you know which strands to choose. Okay? It's going to recognize, it's going to do it in single-stranded DNA, and it's helping separate. As it's helping separate, it's going to add supercoils to the DNA that are going to be very tight, and they're going to be positive supercoils. And therefore, as the helicase is moving in this direction, it is tightening the DNA in the direction that it's moving. That is where gyrase comes into place, by adding negative supercoils. And topoisomerase, comes into place by adding negative supercoils. So as I said over here, it requires ATP to make sure that it hydrolyzes, and it really moves very fast. I thought I had it in my new notes, but I took it out, how many revolutions per second it's actually doing. Now, let's look at the replication fork as this is a bit of a review for you guys. So as we mentioned, first of all, what is wrong with this picture? Look at it for a second. Go ahead, Chandra.
The helicase kind of looks like it's bound to two strands instead of just the one.
It doesn't look. That's how they do it. Yeah, so you're absolutely right. The helicase is bound to a double-stranded DNA. That's not what's happened. So keep an eye on those little details because, as you see, a lot of the times the images given by the artist are there just as an illustration, not to be accurate scientifically. Georgina.
If you go back to the picture, I think that the helicase, it's opening. It's not the polymerase, right? So
No, the helicase is opening. Yeah.
And it's going in the direction of 5 to 3. So the polymerase goes the other way.
No, the polymerase is polymerizing
5
to 3.
So it's making it go 5 to 3.
No.
This is showing it going to 3.
So the leading strand, which is the one here on top, is going 5 to 3. So the polymerase in the top strand is going to be in the same direction as the helicase. The polymerase going in the lagging strand, which is the one that the helicase is in, is going in the opposite direction. Yeah.
So helicase is on the lagging strand?
Yes. Mm-hmm. That's the other way to think about it. Helicase has to go in the lagging strand. Now, so the helicase is going to unwind the DNA that is going to generate those tension upstream of the replication fork. These little dots over there are the single-stranded DNA binding proteins, which are preventing from generating. And what I want to show you here, it's that primase have put a primer over there, and now you have the polymerase 3 putting DNA. Here in the top is the leading strand, and in the bottom you have the lagging strand. And the polymerization of DNA is happening 5' to 3'. So the problem is the lagging strand, as you know. The lagging strand is going to polymerize in the opposite direction, away from the fork. Whereas the DNA in the leading strand the polymerase is basically as Georgina said, it's following the helicase in the leading strand. The lagging strand, the polymerization is happening in the other direction. So what is going to happen is that you're going to now have the formation of the Okazaki fragments, which are going to allow for the smaller pieces of DNA to be replicated. What is not shown here very accurately is that the helicase and the primase are actually associated together, because the helicase is in the lagging strand. So as the helicase is opening the DNA, the primase is putting a primer. Now, in the lagging strand, you have that discontinuous DNA replication. The primase is going to put the 11 base pair primer. The polymerase now is going to come and use the 3' hydroxyl group of the primer to initiate replication. It's going to continue, and when it finds the primer, it stops. At that point, you have generated the Okazaki fragment, which is that DNA RNA hybrid together with the DNA of the other strand. Now, this polymerase 3 cannot remove the primer. It doesn't have 5' to 3' exonuclease activity. It can only repair mistakes by going backwards into 3' to 5' exonuclease activity. So there is a difference with the polymerase. As I mentioned, the primase is an RNA polymerase, and being an RNA polymerase, it can put nucleotides no matter where. DNA polymerase cannot do that. They need to have a primer. So the RNA polymerase can put nucleotides and make a little piece of RNA no matter where in the chromosome they can find. Yes, Chandra?
What exactly is the mechanism of that? Like, why is it that the RNA polymerase can do that while DNA polymerase?
I'm going to try to answer that question in a little bit. So hold up for that. Now, so let me just give you the indication here. Here is an electron transmitted microscope picture of a little chromosome that is being replicated. Notice the status structure. So here you have the circle and the division in the middle. And I highlighted in orange for you the replication forks, so you can see them. So this is an image of the chromosome division that we can actually see experimentally with the microscope, because the DNA molecule is so large. So remember, from the original replication, you're going to have a leading strand to the top, a lagging strand to the bottom for the right replication fork, and for the left replication fork, the bottom strand is the leading strand and the top strand is the lagging strand. So remember that because of the directionality. The top strand is the 5' to 3', 3' to 5', the bottom strand is the 5' to 3'. So this replication fork is moving to the left, and this other replication fork here is moving to the right. To deal with those Okazaki fragments, once the DNA polymerase 3 reaches the primer, it will fall out. It found a block and it just falls off. And here is when now you have another complex, which is going to be the DNA polymerase 1 and the ligase that are going to come into place. DNA polymerase 1, as I mentioned, is only one gene product, and it is able now, because it has 5' to 3' exonuclease activity, basically it's cutting as it's moving forward, it's removing one by one the nucleotides of RNA of the primer, and in their place putting a DNA nucleotide. So as you can appreciate from this image, eventually the DNA polymerase 1 completely moves and removes the entire primer. This image is deceiving because it looks like the primer was taking off like a block of Lego. It's being taken off nucleotide by nucleotide as they're being cut out, as another nucleotide is being added in their place. Now, you have now a gap in between the two DNA strands. You have a free 3' hydroxyl group in the end of this DNA molecule and a free 5' phosphate at the beginning of the other molecule. That gap needs to be fixed, and that gap is fixed by ligase. So ligase is going to be able to fix that nick to now give you a complete, beautiful, continuous DNA strand. Okay? when you look at the replication fork as a whole, it is put together, as they call it, in a replisome. So my question for you guys goes out. What is the problem with this image here again? Why is this picture incorrect?
Because
Yes.
the polymerase is on the wrong side. Isn't it supposed to be on the lagging strand? The helicase is.
Helicase, not polymerase.
But I mean, like, yeah.
Yeah, the issue again here is the helicase. They put the helicase in the leading strand. Like the artist whoever didn't check that out. So no, part of the issue is that you remember that you have tau, here's the molecule that is keeping the two polymerases together. I'm going to load for you a movie that is going to show you how the lagging strand is constantly being released. The leading strand is being synthesized in one sweep motion. The lagging strand has to be grab, synthesize, release. Grab, synthesize, release. And that happens in the replisome. So this polymerase over here opens up and releases the lagging strand, so it can now, notice over here where my mouse is, you have primase putting a primer there, and now you have single-stranded DNA proteins. So here's the previous primer. Here's the previous primer, and the polymerase is synthesizing to the left here until it finds the next primer. In order for the polymerase now to begin synthesis here, it has to let go of this strand and pick it up right here where the new primer is. So there is a motion of letting go and picking the lagging strand to allow for the replication for it to happen forward. So the complex of the replisome, you have the 2 DNA polymerase enzyme, and this is only at one replication fork. So you remember you have two of those. You have primase there, you have a helicase that should be in the lagging strand, and the ligase helicase complex, and the primase helicase complex opens up the fork and adds a new primer to be used in the lagging strand synthesis. So you just have it here. Now this picture is from a different book. I like it better because it shows you the flipping around of the molecule. Here you have, there we go. Here you have now gyrase. It's there somewhere along the line, unwinding the DNA that has wound by helicase. Here you have helicase and the primase together, putting that little RNA primer here in the way. And here you have the sliding clamp and the polymerase that are going to do synthesis. Lagging strand is at the bottom, leading strand is at the top. The replication in this case is happening to the right of the image. The gyrase could be anywhere, by the way. It doesn't have to be placed in that position. As long as it is unwinding the DNA on a circular molecule, it doesn't matter where it's doing it. So you have the single-stranded DNA binding proteins binding there to prevent the reannealing of the strands that are being separated by helicase. You have the primase putting the primer that are eventually going to now be used by the polymerase to synthesize the new strand. Shown over here, you see the little primer. Here's the new strand coming up, moving this way, and eventually it will find the primer again. Once that it's found, it lets go and picks up the next primer together. This is a little movie that I'm going to upload for you later. It's going to be really cool. You're going to see it. And the DNA ligase and polymerase 1, as you see, they're here as a complex. So as the DNA polymerase 1 removes the primer and puts the DNA nucleotides, eventually the ligase finds the gap and seals it. So this beautiful machinery works in awesome coordination to make sure that the chromosome is replicated with a lot of fidelity. Now, gyrase, as you know, breaks down the double strands and is able now to unwind the activity because as the polymerase and helicase are going into the direction of the replication fork, it's overwinding the DNA. And the DNA that is overwound is going to be so tight that eventually the enzyme is not going to be able to separate it. So you need to relieve that tension. That is what gyrase is doing in that case. So It's what it's saying is that it's going to use the ATP energy to make sure that you have the unwinding of the DNA and therefore allow replication to continue forward. That's fairly straightforward. Any questions about this part of the material before I go to the next jump? Some of this is similar to what you learned in Bio2, except that now you have the names, the right names of the enzymes and stuff. Like I'm teaching Bio2 right now, and they call that the repair polymerase, the polymerase 1. It's like, no, that's not what is happening. Anyway, all right, let's talk about this DNA polymerase 3, because I want to talk to you about the issue of what it can do. It is a complex of proteins, and we can consider it by looking at 3 subunits that, or subassembly, excuse me, that they can have. The first subassembly is the alpha, epsilon, and theta subunit that has the DNA polymerase, the 3' to 5' exonuclease, and this epsilon exonuclease. And we call that the core enzyme. If we were to put together this enzyme on the lab, those are the 3 things that we'll need to have a functional enzyme. That core unit. Now, as we mentioned, it needs to dimerize. So if you want to dimerize it, we can add now this tau subunit, which is going to allow it to dimerize. And that is now we call now the polymerase 3'. So now you have a dimer. But that doesn't make much of a sense without the capacity of the enzyme to bind DNA or this other complex with the sliding clamps to keep it in place. That is now called the gamma complex. Together, all the proteins put together are called the DNA holoenzyme. So when you think about the word holoenzyme, it is always considered that it's an enzyme with every single one of the subunits that it needs. The core enzyme is the minimal requirement of an enzyme to do a job. And that's important because we're going to be looking at that in a moment when we look at the RNA polymerase, especially with bacteria. Now, the enzyme has this 3' to 5' exonuclease activity. That is what gives the proofreading capacity and allows for the high fidelity of replication to happen. And the best way that I can explain that is as if you think of the enzyme as having gears. The enzyme is going really fast, putting nucleotides in the replication strand, and eventually it's going to make a mistake. It actually makes a mistake every 10 to the 9 base pairs. Now, actually that's not correct. It actually makes mistakes every base in a million. The fact that you have a way to fix those mistakes is what is important. So now, here for example, in the DNA strand is being replicated in dark green. The light green is the template. You have an A that needs to be read to add a T, and the enzyme added a C, creating a mismatch. The mismatch creates a bubble. That bubble gets detected by the enzyme as a conformational structure. The enzyme goes in reverse, going now 3' to 5'. Going backwards, it can remove nucleotides. And as it removes the nucleotides, it can put it back into forward motion, and it goes back to adding nucleotides. So that mismatch removal brings down the fidelity of the enzyme to 1 error every 10 to the 9 base pairs. So really a fast way of replicating. So mutant enzymes that do not have that 3' to 5' exonuclease activity, they are very error prone. HIV polymerase doesn't have that. Therefore, it's making many mistakes which the virus in HIV uses as a way to drive selection. Because it had the mutation, therefore it can get advantages against drugs, for example, that we're using to treat HIV. Now, let me put a conundrum here for you guys. That enzyme, it's working really fast to divide the chromosome, to replicate it. And in the lab, we had to say that a speedy E. coli is going to divide about every 20 minutes. But the enzyme is only able to replicate the chromosome, depending on the strain, in about 40 minutes. I see some papers that talk about it being in 115 minutes that it takes to divide the entire chromosome. So how can a bacteria that is dividing every 20 minutes divide its genome every 40 minutes, for an example? And that's the thing that I'm going to show you right now. And I just found a publication recently from the EMBO journal. The European Molecular Biology Organization has a journal, this EMBO. And I just chose two of the pieces because what the scientists were doing was that they were using fluorescence in bacteria to localize the origin of replication in the bacteria they were looking at. And those are represented by these little green dots. So they hypothesized that if the bacteria it's going to initiate division in a single origin of replication, here you have the initial fork. You can see the little two origins and the forks happening. The zeta structure, you can see now that the replication, the origins are a little bit more farther away. And eventually, when the bacteria separate, now you have the two forks into different bacteria, and the dots are really far away. So what they found is, it's that replication is happening in the bacterial chromosome at multiple times consecutively. So what you have here is that the chromosome is replicated at 4 different origins of replication. So you can see this chromosome is dividing, is almost dividing out, but there was our original division at the origin of replication that gives you 2. So this chromosome, which are still together because they're still being replicated, has 4 origins of replication. One there, one there, one there, and one there. As the molecules are replicated and separated, notice here that now the DNA is completely separated inside the cell, but replication already started, even before the cell has separated. And therefore, when the 2 daughter cells inherit now the new nucleosome, you already have 4 distinct origin of replication in the chromosome. As opposed to eukaryotes that only replicate their DNA in S phase, the bacteria is constantly replicating the DNA 24/7. And in that way, the chromosome is almost ready to separate when the cell is actually supposed to divide and become 2 cells. So I put here the text from the figure for you to read it. The C+D mentioned the time of cell division. So I just wanted to bring this up because this is a mechanism that is very different from eukaryotes in which the way they separate their DNA. DNA in eukaryotes, as you know, only separates during S phase. It's only replicated during S phase in cell division. Okay, any other questions about this? All right. So let's switch gears then, and let's look at transcription. Now you know how the chromosome is duplicated. To study for this, what I want you is to begin to use your reasoning skills and think what kind of phenotype a mutant in any of those proteins in the table will look like. If you have a temperature-sensitive mutant, what will happen if you have a cell that is growing in the permissive condition for DNA A, and now you switch it to the non-permissive condition? What happens as a phenotype for that? So begin to think and investigate what will happen with every single one of those proteins as a temperature-sensitive mutant. So, as you know, since these are proteins which are required for bacterial cell division, if you mutate them, normally they will die because they cannot replicate their DNA. That's why we have to think about them in temperature-sensitive mutants. So think about that. Put together in your mind the way in which the DNA is replicated, and then think from those steps what happens if you mutate some of these proteins. All right, let's then talk about transcription. Let's talk about the polymerase first. The polymerase in bacteria has 5 units. It has an alpha unit. It has beta and beta prime unit. It has this, can you guys in the Greek system help me, the little W? Huh? Omega? To me, the omega, I always think about the capital omega, which is the other one. So thank you. Omega. And there is also a sigma factor. So the enzyme for replication of RNA, for transcription, only needs the beta units and the alpha. So that enzyme will work. So you have the alpha, the beta prime binds to DNA, the beta units bring in the nucleotides to bring them in, and the omega unit, it's helping stabilize the alpha. And you can see that picture here from the book as this structure there. Notice the difference in the archaea and eukarya enzymes. They have actually 12 subunits. This is part of the evidence of evolution that we believe that archaea and eukarya split together as two domains, way after the generation of the bacterial polymerase. Now, the holoenzyme, the enzyme that it's required for its proper function, has the sigma factor. Now, that core enzyme can bind anywhere in the DNA. It doesn't have any specificity for the promoter. The sigma factor gives a specificity for the promoter. So the holoenzyme knows where the promoter is. The core enzyme will start to bind DNA at random places, sort of like the primase, and start making RNA. So you're required to have the sigma factor. So the sigma unit is required to find where the promoter is and initiate it. And we're going to look into that in a second. Yes, Chandra?
Is the primase basically just the bacterial RNA polymerase without the sigma factor, or is there other things?
It's another completely different gene product.
Completely different.
Remember that in the gene product, I show you that the primase only has one gene product. Yeah.
That's right.
Yeah. No.
Is it structurally similar to the two of them combined, or not even?
I don't think it may be structurally similar, but I don't think that it has all the other capacities to do this. Because it doesn't stay. It falls off after 11 base pairs. This one stays until it's told not to. Yes, Yanni?
Do bacterial RNA polymerases have multiple sigma factors in order to locate different?
We're going to look into that because that's actually true. There's about 8 sigma factors that we're going to look. And those of you who took genetic only learn about one, sigma 70, most likely. So we're going to look about the other ones. Now, what we're going to show you is the issue. Why is the sigma factor important? They did an experiment that I'm going to describe to you. The experiment is to take the enzyme and separate it in a column and then add back to see how binding the DNA binding could be. So here is the experiment. The experiment is done with radioactive tritium, and you have a filter here where you have your double-stranded DNA. The double-stranded DNA, you can add it, and then you can see it can go across the filter easily. The protein, however, cannot go across the filter easily. So what they're measuring is, would the enzyme, either the holoenzyme or the core enzyme, if added, retain the DNA, as an indication of how tightly that enzyme binds to the DNA. So here's the protein-DNA complex. The hypothesis is they will stay together like this, and therefore not see. So you're now measuring the amount of counts in the filter. So what the data show is that if you add DNA to the core enzyme, you're going to have an initial binding, but eventually the DNA is going, with time, the DNA is going to pass through. So you're going to lose your radioactivity in the filter. But only when you add the holoenzyme do you have a stable DNA protein complex that doesn't disassociate. And that is, the idea is that it's required from the sigma factor to give that protein. So let's take a look then at what the sigma factor may be binding. When you take bacterial genes and you align their sequence of the promoter, you begin to see that at position -10 of the DNA and at position -35 of the DNA, they seem to be some conserved sequences. Those are the ones here shown in yellow. They call this area in the -10 the PRIP-now box, or some of the people call it in eukaryotes the Tata box, because it has TA as a repeated domain. The other side is the -35 domain, and that is a little bit more upstream. So you see now that every bacterial promoter has a particular signature, and the rest of the promoter doesn't matter. It doesn't matter the spacer in between the -35 and the -10. These are numbers which are more or less, because as you see, the spacer could be 15 or 17. So it's more or less minus 10 to minus 35. It's not exactly that, especially the minus 35 bucks. Here you have the first base. That will be the first base of the RNA molecule. That doesn't mean that it's the first base that is of the protein encoding molecule. As you can see from this image, you have what is called the 5' untranslated region, UTR. That means that there's a region of RNA that doesn't get translated into protein, but is required for protein stability, for RNA stability, excuse me. And the last part will be the codon that is here shown as ATG, that, as you know, encodes for a methionine. And in bacteria, every protein begins with a methionine. So that is part of the recognition of where, it's part of the recognition of the area which is the ribosome binding site. So the ribosome knows where in the RNA that it made to start synthesizing the protein. So now what is shown is that the sigma factor recognizes those minus 10 and minus 35 regions. The holo enzyme, because it has the sigma factor, can see now the promoter. The core enzyme with the sigma factor can bind anywhere in the DNA and start transcribing, excuse me. So the sigma factor confers stability. So now this leads me to the question that Yanni asked. Here I have in this table a picture of the 1, 2, 3, 4, 5, 6, 7. I said 8, so here I'm only having 7 sigma factors found in bacteria, in E. coli. Next to the name of the sigma factor, you have the name of the gene. So the RPOD gene is the sigma 70. That is the most common sigma factor, and is going to be present in most of the genes that you and I have studied. The other sigma factors are initiating transcription under special circumstances. For example, sigma 32 have a different recognition sequence in the upstream, so the minus 35, and is looking to turn on genes in response to heat shock events. Misfolded proteins in the periplasm, if they're found, that turns sigma 24. Stationary phase. As the cell goes to primary and now it hits stationary phase, it's going to activate genes via sigma 38. So different sigma factors are required to turn on genes that may be required at specific times or conditions. Let me show you this experiment here. This experiment here has the activation of genes by different sigma factors. So I put the sigma factor names next to their genes because the table is listing them all by genes in here. And it's investigating cells in the upper table here, upper graph, excuse me, in growing phase, and in the lower graph, cells that are growing now in stationary phase. So we're looking at two different phases of the cell cycle. What you can appreciate is that there are a lot of genes which are binding to the sigma factor 70, both in this growing phase and stationary. The sigma factor 54 is there, some of them can be bound, and the sigma factor 28 also present there. But notice that during the growing phase, there is no binding on sigma 38, which is the S phase sigma. Only cells in S phase can now show binding of the sigma 38, which is the blue bar over here. So that gives you specificity of when the sigma are able to initiate transcription of groups. And one of the things I want to point out to you is the concept of the regulon, which is right there. The regulon is the ensemble of genes that are controlled by a transcription factor. So all the genes which are bound by the sigma 38 that are turned on during stationary phase are part of the stationary phase regulon. So all the operons bound to it are part of that regulon. Okay? So now, we have sigma factor bringing the holoenzyme to the promoter. That initiates transcription. Let's take a look at what is happening over there. So the polymerase, the RNA polymerase, is able to bind DNA. It's again moving for, the RNA is going to be made 5' to 3'. So it's synthesizing from the 3' to 5' strand, from the lower strand. If you think about it, if you put them 5' to 3', so it's reading the lower strand. And as it is doing, it's creating a bubble that is going to allow the enzyme to bring nucleotides and now synthesize the RNA. That unwinds the DNA about 2 helices. So about 20 nucleotides is all the unwinding that is needed. Not sufficiently to require gyrates to come in to save the day, to unwind it. And that transcription bubble goes really quickly. Now, to initiate transcription, the holoenzyme comes together. So here's the polymerase, the core plus the sigma factor. They bind together. They recognize the promoter. You begin the initiation of transcription. After about 6 nucleotides, sigma says, so long, I'm out of here to initiate transcription again somewhere else. And now the core enzyme can continue transcribing the gene and making the RNA molecule without a problem. When it gets to the end, it falls off. That leads to this kind of image, where as you can appreciate, this is one entire gene right here. And if you look, that gene is being transcribed. So this, if you imagine this as a feather, the little short part are tiny little pieces of RNA being made. And as you move down the gene, those strands become longer and longer because you have had a lot more RNA transcription. But as you see, one RNA polymerase doesn't wait to finish transcribing for the next one to begin. The moment that the polymerase have moved out of place, another one comes in and begins transcribing immediately. And therefore, you have a choo-choo train of polymerases that are making RNA along the gene, making a lot of RNA out of one of the operons. Okay? So that happens all simultaneously. The polymerase doesn't wait to finish transcribing before it initiates the next one. Yes, Reed?
There are different amounts of speeds that you can do that at, right? Like what regulates that?
Well, the speed of the transcription is going to depend on how easily the molecule can fall off at the end.
Well, I mean like the distance between that. Like you're not going to have a million or so transcription.
It's most likely going, I guess what you say, it's most likely going to be the distance where another polymerase can happily bind to the promoter.
Okay.
So it's going to be that. And I don't know how big the polymerase per se is. But as I showed you here earlier, the moment that the polymerase begins transcribing, 6 nucleotides are being added, sigma gets out. So sigma can bind to another core polymerase to begin binding at the promoter and initiate now transcription down the line. So how is this stopped? How does the polymerase know where to stop transcription? You need to terminate it. It's an active process of termination. And termination happens through termination loops. At the end of every gene, there's going to be a termination sequence. That termination sequence contains a pair of inverted repeats. As you can see, those inverted repeats are shown here in red. When the RNA molecule is made, now you have those inverted repeats also in the RNA molecule, which are going to allow it to form a loop. That stem loop, it's the signal that is going to give the RNA polymerase the stop command. But not only that, notice that there is a poly-U tail to that loop as well. So together, that loop and the poly-U tail are going to be important for stopping the reaction. And the termination can happen in 2 different ways. One of them is going to be rho factor independent. And let's look at that first, which is basically the stem loop termination. So this termination, you have that really long chain of U's. Remember, U's bind to A's. That's only 2 hydrogen bonds between them. And just like the AT-rich region, it's a region that is melted easily. So when the polymerase comes in here and continues transcribing the RNA, you're going to generate the position of the loop. That is going to make the polymerase stall. In the inside of the replication bubble, you're going to have that U-rich RNA to DNA hybrid, which is not very stable. And that is going to force it's basically going to help the polymerase dissociate from there. So having that loop and the poly-U tail helps the polymerase falls off. That is going to be helped by this protein called NOS-A that is going to help hold the polymerase by the loop. So that UA region, it's so U-rich that it's not going to make a good RNA to DNA hybrid, and that is going to melt, separating the strand of RNA from the DNA. That is Rho factor independent termination. The other one is the Rho factor dependent termination. And the Rho factor dependent termination, it's similar in the fact that you're going to form also a loop. But when you have Rho factor dependent termination, there is a Rho recognition site in the DNA that is going to be transcribed into the RNA as this Rod site. That Rod site is going to allow the binding of this protein Rho. Now, Rho is a helicase, and it is a DNA to RNA helicase. So it's going to be able to start spinning like helicase all the way down to the 3' end of the RNA molecule. So it's spinning like gangbusters all the way down. It finds the loop that has stalled the RNA, and literally it bunts the polymerase out. So when I've been wanting to punt my cat, I think of Rho doing it to help me punt the cat away. So the polymerase pauses because of the stem loop structure, and then Rho comes on, catches up, and bunts the polymerase out. So that is the last part of how termination. So most likely what the book is going to mention is just Rho independent. But when you start doing your own research and reading papers and in other classes, you may be exposed to both Rho dependent and Rho independent termination in bacteria. So I'll end up with this last slide here. And that last slide, it's the general explanation of the operon in DNA of bacteria. So as opposed to eukaryotes, which have a monocystronic organization in their genes, basically one gene gives you one RNA molecule, the genes in bacteria, remember that the chromosome is very compact, and you have about 88% of that chromosome encoding for protein. How is that possible? We know already that it doesn't have introns, so you have eliminated a lot of that extra material. So to make things more easy for the cell to respond to it, the genes have been organized in operons, and this organization basically put multiple genes under the control of one regulatory unit, which is going to be the promoter. So all the operon genes, in this case, this operon has 3 genes, gene 1, gene 2, and gene 3, kind of like the beta-galactosidase operon that you have learned before. And it has one promoter shown here by that little funky looking red arrow. You also have a leader sequence that eventually that's going to be made into the RNA. And this brings all those genes physically together in the chromosome, so they are all contained under one area. They are regulated all at the same time, so you do not have expression of those genes differently as they're going across. And it's a really common way for the bacteria to turn on genes rapidly to adapt with their environment. Think about the lac operon that we have discussed. When lactose is present, all of them get turned on immediately to allow lactose transfer and lactose breakdown into glucose and galactose. So as the operon gets transcribed, now you have one polycistronic RNA molecule. One molecule that has multiple proteins that it can encode. And here is when the interesting thing happens, because this polycistronic RNA molecule has what is called different ribosome binding sites. So you can have a ribosome binding site at the beginning that is going to produce the open reading frame 1, so you have protein 1 being made. Now you saw that little line is going to be the other ribosome binding site. So now another ribosome can bind and make the protein 2 from open reading frame 2. And last but not least, you have another ribosome binding site here that can make the third protein. So you have this internal ribosome binding sites that could be used to make multiple proteins down. Anyway, with that, I'll finish the lecture. We're a little bit early, so you can have time to do other things. And remember today in discussion, get your phrase scantrons to bring it in, okay? I'll be posting a lecture online for Tuesday of the next week. Thank you very much, guys.